feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Loo School of Medicine Healthy Longevity webinar. Thank you for joining us today. Dr. Nicholas Stroustrup is our guest, talking about a very important topic dear to my heart, and that's systems biology. Please use the Q&A function to send your questions, your remarks, your comments. As always, we first start with a short presentation, and this time by Anna Such. She is a research fellow at the Yong Loo School of Medicine at the National University of Singapore, and she presents her work about personality profiles of middle-aged and older suicide attempters.
Thank you so much. So, as mentioned, my name is Anna Such. I'm a research fellow at NUS, and I'm going to talk to you about this project on personality profiles of middle-aged and older suicide attempters. So, suicidal behavior in the second half of life is quite tricky to prevent, and clinicians have been struggling to identify vulnerable individuals early on, so before these individuals actually find themselves in a suicidal crisis. So one reason behind that is that suicidal behavior in the second half of life is very diverse, and there are likely more than just one vulnerability profiles underlying it. So in this project, we investigated this from the standpoint of personality, and we wanted to find out whether middle-aged and older suicide attempters had different and clinically relevant personality profiles. So in order to do that, we used a sample from the longitudinal research program in late life suicide from the University of Pittsburgh, where we had 285 participants with a mean age of 63 who had filled out the relevant personality assessments we needed for this project. From these 285 participants, 109 were suicide attempters, and we used the data of these participants in a latent profile analysis on personality traits to identify attempter personality profiles. We then compared these profiles to depressed comparisons and non-psychiatric comparisons, who were the two other types of participants in the sample. So you can see the 7 personality traits we use for the latent profile analysis on this radar plot. And you can also see these two shapes. The dark gray shape is the non-psychiatric comparisons, and the light gray shape represents the depressed comparisons. And these are the 3 profiles that we found. So we found a profile we labeled careless labile because they were very high on neuroticism, which is the personality trait of experiencing negative affects often. And they were fairly low also on conscientiousness, the personality trait of being dependable or detail-oriented. Then we had a profile we labeled rigid extroverted attempters, and these people were very high on agreeableness, very high on extroversion, and very high on need for closure, which is the personality trait of needing black and white answers in one's life. Then we had a callous narcissistic attempters profile, and this profile was very low on agreeableness and very, very high on grandiose narcissism, which is this personality trait of having an inflated ego and feeling one is better than everyone else. So these profiles also helped to, or basically differentiated the attempters on clinically interesting characteristics. So here we have suicidal behavior characteristics measured cross-sectionally, and we can see that the rigid extroverted attempters, so the yellow group, they were actually older by about 20 years on on both their first suicide attempt and on their most recent suicide attempt. Whereas the green group, the callous narcissistic attempters, they were actually at a higher level of intent and planning at their most medically serious suicide attempt. In terms of other clinical characteristics, the careless labile attempters had the most borderline personality pathology of all groups. And together with the callous narcissistic attempters, they also reported more childhood trauma. The rigid extroverted attempters had lower cognitive functioning, both at baseline and also a steeper cognitive decline at 2 years follow-up than most other groups. So basically, we managed to identify clinically relevant personality profiles within attempters, showing that attempters are a heterogeneous group in the second half of life. And this has maybe also broader relevance for suicide prevention, where personality assessments could be maybe considered more as a routine assessment in clinical settings. So I would like to thank all my mentors and collaborators for this project. And here is my email address. In case you have any questions, please reach out. Thank you so much.
Thank you so much, Anna, and especially always attempting us to have a discussion about personality traits also in the field of healthy longevity. I think it's very important, so thank you. Over now to systems biology. Dr. Nicholas Stroustrup trained as an electrical engineer at Princeton University and received his PhD in systems biology in 2013 at the Harvard University. He became an independent research fellow at the Harvard Medical School in 2013 and became a group leader at the Center for Genomic Regulation in Barcelona in the lovely country Spain in 2017. And to lay the foundation for future therapies against diseases of aging, his group develops and applies new methods to measure, model, and control, hopefully, the organismal dynamics of aging systems. It's lovely to have you here, Nicholas. Over to you.
Hi, thank you very much for hosting me and for the nice introduction. So today, I'd like to talk a little bit about our ongoing research. Let me share the screen. So today I'm going to tell you about some of our recent work on the molecular genetics and systems biology of aging. So as we age, we experience a stereotyped set of physical declines that move us from youth into old age and death. And remarkably, there's no set rate at which these declines occur. Each individual ages at their own speed, ultimately producing a wide distribution of death times. So aging makes us worse off, but it also makes us different from each other. If you look within age synchronous cohorts, young people are relatively homogeneous. You can see much more heterogeneity in old age. So aging is a process that generates heterogeneity between people. If you ask people on the street to explain why this heterogeneity arises, they'll give you 3 reasons usually. Genes, lifestyle, and luck. The nice thing is today we don't need to take surveys. We actually know quite a lot, at least in general terms, about why some people live longer than others. So over the last 20 years, there's been several twin and genealogic studies that put a maximum on the heritability of lifespan at about 20%. Which means 80% of the individual variance that we see is not the result of variance in our genes, right? So it's probably not our genomes that are driving this, why some people live longer than others. It's hard to control for environment in humans, but you can do so in laboratory animals. So here I show you the survival curves comparing humans and C. elegans. On the x-axis, there's the age, and on the y-axis is the fraction of individuals that age that remain alive over time. And so in C. elegans, you can take animals out of the wild and put them into controlled laboratory environments, and you change their lifespan. But as you can see, you don't necessarily decrease lifespan variance by that much. So it doesn't seem like diet, exercise, or other lifestyle choices are a major source of lifespan variance in animals either. So variance appears to be intrinsic to the aging process, generated by the underlying molecular biology. And so my lab studies the molecular genetics of why this might be. So I'm going to tell you about a particular project that we recently had published over the summer involving an approach we called async-seq, where we're taking advantage of this heterogeneity between old individuals, which we call population asynchrony, to systematically map gene expression networks and understand the mechanistic origins of lifespan variation. So at the core, async-seq is built off of a transcriptomics technology that allows us to collect all of the mRNA from a single individual. So we can start doing population scale RNA sequencing. And so through technical optimization, we managed to get the price of a single worm's transcriptomes down to 18 euros per sample, which means we can start looking at very large populations and look at the distribution across the transcriptome of each gene's abundance. So we can start at the molecular level across 10,000 or more genes. We can start measuring very precisely how individuals are differing from each other. And this is the work of a talented team in my group, including Matthias, Ali, and Natasha. So ideally, the experiment we'd want to do, if we could, is to basically produce data that matches this cartoon that I'm showing you, which is for every gene in the transcriptome to map its trajectory throughout aging and see how each individual, each line here, has a different trajectory. Unfortunately, we can't access this data because collecting a transcriptome is destructive. We can never go back and measure the same individual twice. So what we've decided to do is adopt a statistical physics approach that lets us sample cross-sectionally from this large population of individuals and start studying the distribution of gene expression states, not continuously throughout life, but comparing a group of animals young and old. And from this distribution, we can describe what we see as the mean expression for each gene, but also its variance, which is how variable is this gene across a population of young or old individuals. So here I'm showing you the data from an atlas we produced. Each point is a gene, and for each gene we've measured the mean abundance on day 8, which is a fairly old worm. And on the y-axis we measure its interindividual variation. So if you've ever measured lifespan variant transcriptomic data before, or worked with a data set like this, one of the remarkable things about genes is they're expressed at widely different abundances. So the most abundant gene in our cells and in C. elegans cells is millions of times more abundant than the least abundant gene. So there's a wide dynamic range. And when we started for the first time looking at variance, we actually saw a very large dynamic range in the variance of genes. So some genes are very homogeneous across the population. All of the animals look identical. But in respect to other genes, there's very high variance. And so we were excited to see that there's a lot going on across the transcriptome. So then you can ask, well, how does aging affect this variance? And so we saw, as you can see here, there's genes in red that go up with age and genes that go down with age. And this has been studied quite extensively before. But what we saw, which is very interesting, is a set of genes that whose variation go up with age, but don't change their mean abundance. Right? So most of the transcriptome, as you can see, is going up in abundance, going up in variation. Individuals are becoming more different over time. But there's a set of genes where the population is drifting away from the population average. So each individual is quite far away from what you'd consider a typical individual. But this is actually almost 40% of the genes. So almost half the transcriptome individuals are really very different from each other in old age. And this is a set of aging phenotypes at the molecular level that I think most people would have missed or not appreciated. If you do bulk transcriptomics, which most of the C. elegans work has been done before, you'd miss it because you average out over lots of individuals. If you do clinical studies, I think this type of interindividual variance would be attributed to genetic environmental factors, when really, at least in C. elegans, it's generated spontaneously in a homogeneous environment. And finally, single-cell methods are going to conflate differences between cells within the same individual with differences between individuals. So this is kind of a very interesting aspect of aging that we hadn't been able to measure before. So gene expression increases dramatically with aging, but we wanted to understand where this noise is coming from. Right? So C. elegans, like in humans, has differentiated tissue types. It has neurons, it has the skin, the hypodermis, muscle, intestine. And what we can do is we can start looking in our across the population how genes are correlated with each other. So what we found when you look at tissue-specific transcripts, genes that are only expressed in a single tissue, you see that all of the genes expressed in the germline co-vary. So here on the X and Y axis, you have all of the genes, and then the points are colored by how correlated they are. So you see this big red block in the germline, which means that all the germline genes are up or down together across individuals. And this is anti-correlated with most of the genes in the soma, the hypodermis, the intestine, the muscle, which are then correlated with each other. So it suggested that there's something going on at the tissue scale that's producing this gene expression variance with age, right? And so another way to look at this is if you look at the correlation, the distribution of correlations across all gene pairs, you can see this bimodal distribution. On the right, there are genes that are highly correlated with each other, which are genes in the same tissues. And then you have, on the left, you have genes that are anti-correlated with each other, which is the genes that are expressed either in the germline or the soma, and these are anti-correlated. Right. So when we started to look about where this variation is coming from, we figured out a way to measure in absolute terms the total size of a transcriptome of animal soma or in their germline. And what we saw is in youth on day one, some individuals are larger than others, some individuals are small, but most of the individuals clustered around this line, which suggests that animals have some sort of mechanism that's very carefully regulating the size of the transcriptomes in the germline and soma. So animals are kind of nicely proportional in youth across their organs. When you fast forward to the end of life, you see a much larger spread. So the germline gets smaller, but what happens is some individuals have large germlines and small somas. Some individuals have large somas and small germlines. And so it seems like whatever has been coordinating the organ sizes in youth is now broken down in old age. And this is responsible for the gene expression variance we're seeing. And one of the amazing things about working in C. elegans is you can very quickly do interventions to test your model. And so what we can do in C. elegans is we can ablate the germline. So we can create a large population of individuals who now lack this tissue that's varying with age using a genetic approach. And what we find is we completely eliminate this correlation structure in gene expression. And so this demonstrates that it really is this inter-tissue communication breakdown that's generating gene expression variance in old age. Another way to look at that, if there's people who like principal component analysis, is you can look at those sources of variance. And you can see that the intact animals with a germline are spread out over both principal components, but removing the germline compresses the variance and makes animals cluster the small point on the graph. So we were excited to be starting to understand where gene expression variance comes from in aging. And we wanted to then link it to say, well, maybe if the germline is generating gene expression variance, perhaps it's also responsible for generating lifespan variance as well. And so we looked at an experiment, and people have known for a long time. Judith Kindle and others, almost 30 years ago, showed that when you remove animals' germline, you extend their lifespan. But now we measured very carefully to look at gene that lifespan variance phenotypes. And what we found was surprising and exciting, which is that when you remove the animal's germline, you're not just extending lifespan, you're actually reducing variance in lifespan. So animals are living longer and their lifespan is more similar to each other, which is not what you usually see with interventions in lifespan, which tend to keep the coefficient of variation and mean lifespan in perfect proportion. So this is the first the intervention we've seen it done this. And if you run a regression model, the details aren't so important, but essentially what the AFT regression model is doing is it's putting the two curves on the same time scale. So you're removing the average difference in lifespan. And what you reveal is just the residual effect on variance. And you can see that when you remove the germline, you're actually removing about more than half of the inter-individual variance in lifespan. Right. So not only is this tissue scale coordination that we're seeing when you disrupt it, you remove gene expression variance in midlife. You also then are reducing lifespan variance in old life. So that brings us to our model, which you have animals, and they sort of separate out in gene expression into two covariant units. You have the soma and the germline, and they vary as a group. And that seems to be involved somehow in variance in lifespan. Right, so this is our model, and we wanted to bring it down more to molecular mechanisms. Can we identify some of the genes whose function are responsible for either the variation in the soma to the germline or the communication between them or so on. So we wanted to find genes whose activity during life, in young animals going on to old, is generating this organismal gene expression variance that we can measure, and then leading to lifespan variance. So there's many different ways you can start looking for these genes, but we realized that a fast and efficient way to do this is that there should be a relationship between genes' effect on lifespan, on gene expression variance, is that if you knock it down, that'll tell you its average effect, if you just knock it down in individuals. And that should then align somehow with the natural variation we see if that gene's average effect is contributing to individual variance in lifespan. And this is a great realization because we can measure the average effect of RNAi knockdowns across a population very easily with just a small number of samples using transcriptomics. And this wild type inter-individual variation is what we've measured in our atlas already. So we realized that we can start screening quickly for mechanisms by using our atlas to guide a perturbation screen. And this is 200 times cheaper than it would be to screen for lifespan variance directly. So I'm not going to spend much time on the mathematical model underlying here. It's really for specialists. If somebody wants to pause the video later and take a look, you're free. But the idea is we're using perturbations to dissect the covariation matrix of gene expression. And just in case people are wondering, this is not an aging clock. It's a very different way of understanding trajectories and individual variation in aging, right? So we selected 90 genes. We wanted genes that are highly variable in either youth or old age. And we wanted to find genes that seem to have their expression level connected to lots of other genes. Maybe they have a causal role in the hierarchy of gene expression. And so we collected genes that are highly connected in the co-expression graph. And we chose like a moderately sized RNAi endpoint screen of about 90 targets. So we knocked them all down in youth, and then we had animals age without these genes we've knocked down, and then we measured gene expression again on day 8 in old age. And what we found is among these genes, they didn't all have the same effect on the transcriptome. So we clustered them. Here you can see like a dendrogram showing the hierarchical clustering of these genes, and there's at least 7 different classes of genes that are having very different effects on gene regulation. So knowing a little bit about what the genes we picked, we knew they had different functions, and we confirmed molecularly, and we confirmed that their organismal effect on gene expression are also very different. So we run our transcriptome scaling model, which is a mathematical model I told you about earlier. And we could then use that to estimate what fraction of variance in between individuals and wild type could be explained by variance in the activity of each of these genes individually. And we were really excited to find 40 hits that each alone could explain more than 10% of inter-individual variability in gene expression. So these might be the mechanisms that we're looking for. And we're particularly excited to find 12 genes that when we knock them down could explain more than 30% of variance, which is as much as the germline knockout would, right? And so the germline knockout is a big hammer to take to an individual. You're removing an entire organ. And now we were being able to do that with just modifying a single gene, right? So we then started counter-screening our hits for lifespan phenotypes. And surprisingly, all of the genes that we tested that seemed to be related to gene expression variance had a lifespan phenotype. So some were extending lifespan when you knock them down, some were shortening lifespan, but they all were doing something. It seems like the lifespan variance phenotype and the gene expression phenotype are very deeply involved in the aging process somehow. So then we looked not just at mean lifespan, but lifespan variance. Remember that we got into this because germline ablation was making animals more similar in aging. And so what we found is that most of our hits that we identify with gene expression are also reducing lifespan variance. Here's a small endocrine signaling peptide NLB28. When you knock it down, animals live a bit longer, but they have a lower variance, a rectangularization of the survival curve. Here's another gene, SCR1. They actually live shorter, but again, more homogeneously. And so here's a summary across all of our targets. So really excited to start understanding some of the mechanisms involved in lifespan variance. And so we also were wondering, are these just genes that are all phenocopying germline ablation? Perhaps they're all just removing the germline, but through the different molecular mechanisms. And we found that most of them did not have an effect on reproduction. So it seems like on the right here, PCN1, yep, that just ablates the germline. And so it's not a surprise it has the same effects of GLP-1, but there's lots of interesting genes here that seem to be having the effect of germline ablation without actually affecting fertility at all. So we're excited to find these genes. And it puts us in a place where we can really start answering rigorously some very interesting kind of general questions that people have been talking about for a millennium. Like, why do some individuals live longer than others? So at least in C. elegans, we now have a partial answer, which is the intrinsic activity of these genes. That we have these genetic loci who are constant across individuals, and all the individuals are isogenic, but their activity spontaneously generates lifespan variants and gene expression variants during aging. And these don't seem to be the same mechanisms that are determining mean lifespan alone, which people have been studying for a long time. So that's very exciting. And also it's interesting that these genes are not just in the germline or just in the soma or specific tissue. They're distributed across lots of different tissues in an animal. And we can start looking at that. So ever since this publication was out, we've been continuing the work, and we've actually started knocking fluorescence sensors to see actually what the activity of these genes are in worms, work by Lucy and Daisuke in the lab. And so here's this endocrine signaling peptide I was telling you about. It was very variable between individuals, and when you look at the protein level, you also see that there's a huge degree of variance between individuals in this protein. So it's not just a gene expression phenotype. You can relate this to the size of individual's genome. We have another marker that allows us to count the nuclei and the animal's genome. And you can actually see that the expression of this peptide is actually correlated with the germline number of nuclei. So there seems to be some link between this peptide, which is expressed in the skin, the hypodermis, and the number of nuclei in the animal's soma. And this NLP28 expression is predictive of lifespan. Right, so we're starting to lock in these gene expression phenotypes as mechanisms that seem to be interconnected in physiologic networks, in this case, influencing germline, that then subsequently have an effect on lifespan. So I think we're getting into some very interesting, systems biology of aging mechanisms. And we have lots of these targets. So some of them are expressed in HYPE, the top. Some of them are expressed ubiquitously, and especially in the germline. And we can actually start seeing inter-individual variation in the expression of some of these genes in single neurons. Right, so I'm really excited moving forward to start trying to understand how different parts of the animal are aging differently and how these different parts are coordinated in order to determine an individual's lifespan. So yeah, so we can refine our model. Almost done with the talk. The model we had before was that the germline and the soma are separately varying and driving lifespan variation. But now we can kind of explode the animal into many different tissues, right? And in each of these tissues, there's specific targets. We know the names of these targets. We found some of them. We certainly haven't found all of them. But the names aren't so important to the model. What's important is that the origins of lifespan variance, or at least the mediators required for lifespan variance, are not coming from a single causal factor, but they're distributed across the individual. And so there's lots of different parts of C. elegans bodies, and by extension, I would imagine our bodies, that are individually contributing variance in aging and lifespan, spontaneously generating it, and then driving differences in outcomes of aging, right? And I think this is an exciting model. I think it's relatively intuitive, even if the way we got here was somewhat technically complicated. But the idea is by doing cross-sectional sampling and doing careful quantitative analysis of gene expression covariance, we now have a way to systematically identify mechanisms that are causes of differences in outcome and aging, which is the type of differences people are trying to measure with biomarkers or of aging, all different sources. We can now start identifying the causative mechanisms generating differences in outcome using this async-seq method. And I think this has broad translational potential. It worked in the worm really well, and it was great. But the method can be applied to vertebrates now, because in the end, you just need to be collecting the right types of gene expression data and performing the right types of analysis. So I'm excited moving forward to start doing some of these approaches in vertebrates, right? And just to summarize with a picture, we all have the same chronological age. We all start out with that, but over time, we all develop our unique biological ages. Some people have biomarkers that would suggest they're younger than others. And what we're getting into with a study is starting to find the mechanisms responsible for converting our chronological age into our biological age. So I'd just like to thank you, thank the people involved in the study, students in the lab, postdocs. It's a really great team to be working with, as well as our funding sources in Spain and at the EU level.
Thank you, Nicholas. Very nice presentation. May I ask you the first question? Is aging and the heterogeneity of it just luck?
Yeah, but it's luck. I would say there's chance in it, but it's chance like a casino game, where how lucky you get is determined by the shape of the baccarat table or the dices that are weighted or things like this. And so, although there's chance involved, we can start to understand the mechanisms that are generating and shaping this chance, and then start to intervene to change those mechanisms to change your luck.
So it's more gambling, yes?
It's gambling, but we're starting to understand the rules of the game, and maybe we can change them.
Okay. So you said that there is the increase in gene expression with aging. We in the clinical world, we are always talk about compensation. We are compensating for something which is not going wrong. So isn't your finding maybe just a sort of compensation of something which did not go well because you had bad luck?
Yeah, so we see an increase in gene expression variations for sure. And yeah, I imagine like what the nervous system is doing in aging is trying to hold everything together, right? All of your organs need to be acting at the same rates and collaborating just like a team needs to collaborate. And we're seeing that breaking down with age. So my guess is there is something being compensated for. And my guess is what the animal is optimizing is probably not lifespan, right? It's probably optimizing something to do with reproductive fitness, right? Because in the end, the worm's main job is to make more worms, right? And lifespan, how long they live, is maybe important to an individual worm, but probably not directly selected for in evolution. And so maybe these optimization strategies that are compensating different parts of the organ to maximize reproduction maybe have a side effect of shortening some individuals' lives more. And so I think that you might be onto something saying that compensation is involved, and it's probably compensating for one phenotype at the expense of lifespan. And some individuals have to compensate more and thus have their lifespan affected more by these mechanisms. That would be my speculative link to compensation in humans.
So in what is the role then of individual organs in it? So do you think that aging is a driver everywhere, or we already have now, I think, clinical evidence that the pace of aging in different organs is different in humans at least, and that might also the aging process in different organs might drive other organs to age faster or to age slower. So do you foresee a network there?
Yeah, I think that it's, one of the fundamental parts of being a multicellular animal, right, or having many organs, is that all of these organs depend on each other to function, right? So if your liver isn't doing well, your brain is not going to be doing great. If your lungs are doing poorly, then your pancreas is going to have trouble. And so there's a network. Some of it might be endocrine signaling, like resisting mechanisms, but there's also just like a functional dependence of all the different organs. And so the aging of different organs are going to be influenced by each other by direct specific mechanisms, some of which we can now identify, but also just functional dependence. If your blood glucose goes down, then all your organs suffer, and that's going to have some long-term consequence of their organ-specific aging. And so I think this network systems biology of aging is going to be important of understanding ultimately the why and when diseases of aging arise.
Yeah, and picking your brain here, because we try to do that in humans, where would you start? So what is the weakest link if you're looking at the interdependency of the different organs? So where does it start? And is it starting in humans in one organ without any heterogeneity, or does it start with one worm in the brain and the other one more in the muscular system?
Yeah, so it sounds like a Billy Joel song, like "We didn't start the fire," right? It's been going on forever. And so my idea is like, when you look at so here, like what do people who study engineered systems and the failure of engineered systems, how do they approach it? And so the idea is that there's like it would be really nice if there was one organ that causes everything else, right? And it would fit into a nice pathway with a top and a bottom. And my feeling is we're not going to be that lucky, in that at the top, there's something at the top, it goes to the bottom, but then the bottom goes and wraps around and has feedback and influences the top again. And so you start having a dynamical system where all of the parts are kind of in orbit around each other, right? And so affecting one thing will shift the orbits of all the other. And so you start needing dynamical systems models, and they don't tell you where the process starts, because it might not start anywhere, but they'll tell you how, when you change one thing, another thing will affect. And so physicians someday might be able to use control theory to, figure out, if you want to fix the pancreas, maybe you need to nudge the brain and the liver and the heart in a certain direction. And so rather than looking for an origin of aging, maybe you could understand the interorgan dynamics of aging, and that might get what you're looking for.
And how would you study that in C. elegans to help us to eventually bring it into clinical practice?
Yeah, so what we're doing in C. elegans now is we're identifying the mechanisms that we can manipulate experimentally to basically make animals less variable from each other, which means these are the genes, either upstream or downstream from these genes, there's symptoms and causes of aging. So in C. elegans, we can start to turn these into levers, right? So if you have a gene in C. elegans, we now have the technology using protein degradation systems that once you identify a gene, you can rapidly turn it into a drug target, right? And be able to quantitatively modulate these targets, and maybe multiple at a time. My lab's been working on a way to modulate 2 genes quantitatively simultaneously. And so you can start running big screens of like doing dose dependence across multiple parts of physiology, and then measuring across the animal how all the different organs change. And this is where I think it's going to be a really interesting application of machine learning techniques, like probabilistic machine learning, and building predictive models trained on dosage series of complex interventions that are kind of picking up all the latent variables in the system. And they might align to organs, they might align to metabolic pathways, we don't know. But the idea is through systematic measurements, partially using what we've published over the summer, but also using more interventional data, using big robots doing intervention screens, we can build kind of predictive models of physiology at the organ level at the organ granularity, and start building very complex dynamical models. You can ask, if I reduce the function of the intestine, this gene in the intestine, by 15%, what are all the other outcomes going to be across all the other tissues? And then where is that going to put the individual 5 years from now on average? And build these predictive models that you can flip on their head and use the predictive models to say, well, I don't want the person or the animal to be there, in aging. So maybe I should move the system in the opposite direction.
Yeah.
And so it's going to be a combination of really careful measuring the most important parts of an aging body, and then using machine learning techniques to understand how that should then produce outcomes.
Yeah, great. You really nicely showed that germline ablation actually increases the health span and decreases the variability.
Mm-hmm.
If you would translate that into humans, for example, what would that mean?
So obviously we're not recommending anybody start messing with their own germline. There are some historical evidence in psychiatric institutions in the US. There was some systematic efforts to perform hysterectomies on women. It's very unfortunate. There's also in medieval Korea, there's some eunuchs being generated who then lived in relatively controlled environments. And in both these cases, I would say that they're statistically weak, but suggestive evidence that these people without germlines were living longer, slightly longer. So perhaps the mechanisms are conserved between worms and humans that would link germline function to lifespan. There's actually some really lovely zebrafish work going on right now. A colleague of mine, Itamar Harel, has been looking in killifish what happens when you ablate the germline, and it's very interesting and complicated, and the biology is going to be fascinating to learn. So it seems like these mechanisms are conserved, but it's going to be a while until we can translate that into an actionable treatment.
Yeah, fully agree. You had a statement that the variance of gene expression, that it's not a clock, I think there is a huge, maybe also a hype around clocks, biological clocks, to measure how old we are biologically compared to our chronological age. Why couldn't the variance of gene expression not be a clock?
So yeah, so at the core of a clock model, there's like a goal, and I completely support this goal, which is you have this big high dimensional change, and individuals are changing, in billions of different ways. Each methylation site in the genome is kind of going its own direction. And if you collect that data set, you can't understand it, by looking at it. It's not an interesting textbook to read. So you need some way of taking this high dimensional measurement and moving into a low dimensional space. And that's what we're doing. I think that's what everybody's trying to do, and it's what we need to do to understand. I think where the people working on clocks overshoot a little bit is by going a bit too far and compressing this complexity down to a single number. As if we have one chronological age, which we do, we were born at a certain date, then we must have one biological age also. And I think that is like a vast oversimplification of what's going on inside our bodies. And it leads to compelling, but maybe a bit reductive arguments, being like, oh, if I give you this drug, you'll rejuvenate because your biological age, goes, from high to low. And probably what's happening is you're giving them the drug and you're shifting this multidimensional space in a very complex way that you can project down as a single number that's going back and forth. But it's probably not necessarily the most informative or predictive of future outcomes. So,
yeah.
So when I say I'm not doing a clock in a lab, I'm not trying to do that. I'm not trying to reduce the complexity to a single number that then progresses with time. Maybe you can do an organ level clock, and that's going to be sufficient that each organ has an age. Certainly, there's lots of interesting work with blood-based organ clocks and things like being out there. But maybe it's also not that simple, because maybe part of your liver and part of your heart are actually more similar to each other than they are to the surrounding organ. So it might not break down to organ level granularity. And so that's why our analysis is not assuming that there's a fixed low dimensionality. What we're doing is we're using interventions. So you hit the animal and you listen to how it vibrates, what changes in the animal, and then you use that to understand the multidimensional changes rather than just doing a fixed dimensionality reduction.
Yeah. So the first human clocks were trained on predicting mortality. Then we were going into the incidence of disease or other outcomes. The first actually was chronological age,
Yeah,
which
I was going to say.
never really, understood, because, but then it was mortality, and then it was more the health span. Do you see in your C. elegans also the association of gene expression, not only with lifespan, but also with health span? Because you can have the swimming or walking or whatever activity, of course, of C. elegans.
So yeah, so we now have a very good transcriptomic clock in C. elegans made by colleagues, so we can predict remaining lifespan, at least for a population, the average remaining lifespan quite well. And so for I give my whole talk about lifespan variance. I think it's very interesting, but in C. elegans, the remaining lifespan of an animal is very correlated to its remaining health span, right? So like humans, C. elegans, they become, they start out youthful and vigor and moving around and exploring the environment, doing all sorts of things. And then over time, they get slower and weaker, and they eventually spend about 10% of their life immobile, which is about the average time people aren't really walking in humans, to my understanding, right? So it's very similar, and they're very correlated. And so our results, where we're talking about lifespan variation, also holds for health span as well, as measured by behavioral aging. So it seems like the two things are similar enough that the basic mechanisms of generating inter-individual variation are driving both.
Yeah. Please allow me to have one more question before going to Tsing. I think the questions are enormous at the moment from the audience. You had a very nice analogy. You were saying organs have to work together to function well, as teams do. Tell us a little bit about your team, how they work together to produce the best outcome, as organs do.
Yeah, no, I feel very fortunate. So my lab is based in Barcelona, right? And I'm American, right? So coming to start a job at this institute, I had to navigate the culture. And one thing that really helped was to build a very culturally diverse lab. Right. So I think at some point we're generally about 8 people or so, plus or minus. And we come from I think at some point we had 2 Germans. That was like the biggest overlap we had. People from Catalonia, from India, from Germany, from Portugal. And so it's been really great being able to get lots of different types of people. And yeah, so it's very interdisciplinary. And I think that's the hardest thing about running a interdisciplinary lab is each person really has to specialize in one method because you need so many. So we have, one or two computational people who are doing machine learning or statistical analysis. We have the transcriptomic wizards who are doing all the libraries and analysis and things like this. And then we have a lot of biologists, cell biologists, molecular biologists who are doing the experiments and collecting data and things like this. So it's been a really lively place to be for the last couple of years.
Great. Over to you, Ting.
Okay. Thank you for the presentation, Professor Nicholas. It's really enlightening. I'm not really doing the homework now, because I'm newly joined. But there are a lot of questions I want to ask. But Professor Andrea already asked a lot of questions I also have in my mind. So the first question I want to ask is do you have any suggestion for me if because I'm planning to start my own work next year soon. So I see you got your team did the fabulous job work in the homework. So what kind of suggestions you will have for a new student to do the warm job?
Yeah, so for example, a PhD student,
Yes.
my recommendation is it should be 90%, 95% about the people and only 5 or 10% about the topic, right? Because the most important thing to do in a PhD is to learn how to be a scientist, how to do experiments, how to think about answering questions at the edge of our knowledge. And it's going to work much better, and you're going to do much better if you're with a team of people that are supportive and interested and interesting. And so, go to an environment where your peers are fulfilled and excited, and the PI is engaged. And then if they happen to be working on an interesting topic, that's a bonus.
Yeah, true. I feel my colleagues who are doing the warm work, they really love their work, even though it's a little bit tedious than the other individual work. Yeah. So then it is. And the second question is I heard that you are already kind of planning to shift your system to the other tissues, right? So and Professor Adria also asked about kind of it's very tough to do that. And then you also so would you talk a little bit about to shifting the whole system to the other different species, different organisms? You already doing some collaborations with the fish,
Yeah, we're
lab.
so
Yeah.
basically so we're not planning to shift, right? So the idea is, I think that there's going to be most of the experiments on systems biology. It's, at least for the next 15 years, it's going to be only possible in C. elegans, just because, you can go 10 times faster, and so you do much better experiments, right? But at the same time, not everything is going to translate well. And so you need to keep your, as you go big in C. elegans, you need to have a sanity check that what you're doing is relevant. And so the idea is to, in parallel, start trying to work in vertebrates, right? And this is something we're just exploring how to do, basically, after having really proven that it works in C. elegans. And so there might be some mouse work you could imagine, redoing async-seq and building a similar gene expression variation atlas in mice. I'm kind of waiting a little bit for some of that. There's a big NIH study coming out that I'd like to see their data to see, if that's going to be good enough for our purposes or if we need to collect more data. There's also some, some studies in Switzerland that might be interesting. But the general idea is, we don't know a lot about how individuals vary in aging invertebrates, right? There's human data, there's these biobank data, and you can do some amazing amount of work in human biobank, but you can't control the environment enough in order to support some certain types of statistical inference. Because in the end, some people are eating cheeseburgers, some people are eating vegetables, and that's going to spread out the variance. And if you try to track that down to a single genetic cause, you're going to get very confused, right? Yes. So there's really great data for humans, but it's noisy. But in model organisms, there's very, very small population sizes. And so we have like the mouse cell aging atlas. That's really great. I think there's gonna be another one coming through, but there's only like 2 or 3 individuals, right? So all you can make statements is how this strain of mice on average changes with age. And so the idea is like what we could do is like inspired by the successes of async-seq and C. elegans, do async-seq in either fish or mice. Fish are great because you can do interventions faster. But it's just choose your invertebrate, and who your collaborator will be matters a lot more than a lot of other things. So yeah, so the idea is to get a large population of a vertebrate animal, and basically again compare young animals and old animals and get 500 pancreases from 500 animals and 500 livers and 500 brains, and then you could look at the joint distribution of gene expression across these organs and across individuals and start really being able to measure systematically how everything fits together. But we're very much at the beginning of thinking about how we might in practice fund and execute such a collaboration. So,
That's gonna be like a huge metadata.
if there's anybody in the audience who knows somebody in a funding center who could pull us for us or would be interested in working on this, would be receptive.
I think the audience has lots of questions, other questions, which can be found in the chat box. I think I will just ask the first one to Nicholas. Can you see change of variation in expression upon drug perturbation? So did you test it with some geroprotectors?
Yeah, we didn't do drugs yet because I was a little bit worried about differences in the consumption of the drug by individuals. We didn't really know how to make sure each individual's got a consistent dosage, although maybe we were worrying too much, and so we can do that moving ahead. But we didn't just do genes. We for example, changed the animal's temperature. So that's something you can get homogeneous. You can raise all of the animal's body temperature by exactly 5 degrees. And that's in the paper. It basically doesn't change the story. It just, the whole story runs faster at higher temperatures. There's a lot more gene expression noise, but it still organizes into the same groups that I talked about. We also changed the animals' diets. So C. elegans are like amazingly evolved creatures for eating bacteria. That's like what they're good at. And they eat live bacteria in the lab and in the wild. But what you can do is you can UV inactivate their bacteria. So it becomes, the nutrition changes and it also becomes non-pathogenic, and they live 50% longer. And again, what we found was this didn't fundamentally change the relationship with germline and soma. It just extended the lifespan while keeping the inter-individual variance the same. So we haven't looked at drugs, but we've looked at a couple different interventions that you can do environmentally, and they seem to not change the story much, which was a surprise.
And many questions are with regards to the pathways being involved and what kind of genes being expressed, and especially around inflammatory processes and systems. Any inflammatory genes inside your investigations and clusters?
Yeah, so I mean, worms don't have an adaptive immune system, right? But they do have a very, very complex and intricate innate immune response, involving a lot of immune peptides that are expressed in response to specific pathogens. So worms know if they're exposed to a virus versus a bacteria versus, they're very careful about this. And some of these peptides are induced in aging spontaneously or by some of the perturbations we've seen. But we don't see the immune system as being one of the chunks that's independently varying. It seems to be more tied to organs than the immune function of organs. But in the paper, there's like big chunks of genes we see partially independently varying, and you can go and look. It's the last figure of the paper. And maybe some of the immune genes you're interested in are in there. Yeah.
Yes, many questions with regards to the data analysis, and then Xing, you can ask the next question from the audience. About AI machine learning and how you actually approach that, because there's so much data. So what is the role of AI in your group?
Yeah, so I mean, when members of my lab join, I give them a probability theory textbook, right? I don't give them like a probabilistic machine learning textbook first, because I think the foundations of quantitative analysis are in understanding how probability and statistic works, which is boring, but I think it's much more fundamental, and you can take that with you in your career, more than any particular technology. You know, these days you're using transformers or optimal transport or, you know, Gaussian processes, but like 10 years from now, it's going to be completely different. But they're all going to involve your probability theory. So learn that and build your base. Also, you know, linear algebra and partial differential equations. So these are like core undergraduate courses that I think are never going to be as exciting as like an online tutorial for AI, but I think are going to build a foundation that is required to move forward. Yeah, and then really trying to understand the mathematical basis of what's going on. These models aren't magic; they're really clever, but they work for certain reasons. And the more you understand about them, the better. So yeah, so in our cases, I think we're not using, you know, deep learning models now because although we collect a lot of data, we're not collecting like all of humanity's writing over the last 20 years amount of data. So we're a couple of zeros short of being able to fit like truly enormous machine learning models. So you know, basic optimizers for probability, like log likelihood statements about how these genes are affecting each other. Are working for us right now. I think Gaussian processes are going to be very interesting because you don't have to make assumptions on the functional form of underlying things you're looking at. That in general, that's what I have to say about machine learning.
Okay. Would love to work in your group. There's another question from the audience. Really understanding what is the basic unit of our aging process on humans?
I mean, I'm not a doctor, right? So I'm a
You're a
biologist with who knows quite a bit of math and statistics, right? So human aging, what's the basic unit? I think it's the individual, right? It's the individual that has an age. And our organs all are subject to, you know, you could take the best pancreas in the universe and you put it into a 90-year-old. And, you know, I would bet in a couple of years, it's going to be a pretty terrible pancreas just because it's, you know, stuck inside of an old individual. So I think the basic unit of aging is always going to be the individual. And there's going to be some complex relationship between all the organs, but in the end, they're either in a young person or an old person. That makes a big difference.
Good. Ting, your part again.
Yeah.
Any
Yes, like I got like a very interesting question from the audience. Like they are asking, like, do you have any comments on one very interesting like the finding in the Peter Fiddich's like model of aging, the relevant networking stability can explain the Cooper's law of aging in many species and apparently aging-dependent mortality in some exceptionally long-lived animals.
Yeah. So I have not read this paper yet. I must apologize. So I can't comment specifically, but I can talk generally about the Gompertz law. So you saw these survival curves that I was showing, and there's the variance, right? And so one way to think about this variance is to look at the risk of an individual dying over time. So, you know, what fraction of a population of 20-year-olds will die in the next year? And that's much lower than the risk of 70-year-olds. And, you know, in like the, you know, maybe 300 years ago, Benjamin Gompertz is a demographer, and he realized this risk goes up as a straight line on a log linear plot, right? So it's a geometric series, it's exponential increase in the risk of death over time. And there have been many attempts since this discovery to explain the Gompertz law in respect to basic data generating processes. So Gavrilova suggested if you have a large system of redundant components and they're failing, that would generate an exponential increase. So I would say my general concern for all such efforts of making structural mechanistic statements and mapping it to this exponential shape is it's not unique. There's going to be a lot of different models with very different assumptions that can all produce an exponential shape. And it comes from the fact that if you've ever heard, you know, there's a lot of normal distributions that you see around the world, and it's because of the central limit theorem. And if you take the averages of a set of numbers, those averages are often tend to be normally distributed. There's something also going on that generates extreme value distributions, which is if you take the maximum and minimum set of numbers, it generates these shapes of distributions of the long tail. And that happens to look more or less like an exponential increase, consistent with the data. And so anything that's an extreme value process will generate a shape that might be consistent with human lifespan data. And so it's really hard to know which particular mechanism would be doing this. And so that's my general concern about overinterpretation of okay, we can generate a Gompers from a model. Thus, does that mean the model's true? It's maybe not that simple.
I guess this will help a lot, like for this audience, like to learn about like your comments for this publication. And then the other question, like last question we have from our audience is, about like they really want to take use of your fundings. Are we able to measure our own biological clock?
Are we able to measure our own biological clock? Yeah, 2 points. One, why would you want to? Because right now, as of
Or the other question from the same audience. Okay, sorry.
There's not much you can do to change your biological age. You can make your life better by exercising more and eating better and sleeping better, right? But you don't need a quantitative measure to know that you should be doing these things. So that's, why would you want to would be my response.
Okay.
Maybe someday there'll be a product that will, rejuvenate
Maybe
you.
just like
But like right now, I think a lot of people are more interested in making money than really getting to the basis of how well these treatments work. So I would be skeptical of anybody who will, tell you should measure your biological age at this stage in the medicine of aging.
It's a pity that the time is over. I would love to have that discussion with you because we do that in clinical practice and use it as a marker. But I think
doctors for sure, but this is a question from a student, right? Who is going to like change their lifestyle over a number they read on the internet, right?
Yeah, absolutely. Okay, thank you so much, because it was a wonderful discussion. I really learned a lot about the variation in gene expression and C. elegans. Thank you so much. Thank you, the
so
audience.
Much for having me.
Yes, really absolutely. Thank you, audience, for being with us. There are always a couple of announcements. We have a conference end of February at the 26th the 27th and the 28th of February. Unlock Healthy Longevity, a conference in Singapore where we bring together entrepreneurs, investors, scientists, everybody who is interested actually in healthy longevity. So please use the QR code to find more information. 2 days before that conference, we are starting our intensive course about longevity. It's really for healthcare professionals learning how to implement, for example, biomarkers of aging into clinical practice and what to do with it to lower the biological age. We also have the talent incubator. It's open for applications. It's running next summer, 2025. So if you are young by heart, please apply. The next webinar is Miriam Grunewald, the Department of Developmental Biology and Cancer Research, the Faculty of Medicine in Israel, will be our next guest. I will leave you with a final video, and that's about the longevity of certain species. Take care.
How can taking care of a grandchild lead to an unexpectedly long life? Back in 2017, this snow leopard was already 17 years old, the equivalent of an 80-year-old human. His agility had declined, and he had lost the ability to hunt. He spent his days lazily lying around, waiting for the zookeepers to feed him, and had no interest in exercise. The old snow leopard was nearing the end of his life when the staff introduced a young, energetic 1.5-year-old snow leopard into his enclosure. The young leopard would sneak up on the old one and even steal his food. Suddenly, the old snow leopard found himself engaged in daily battles of wits with the youngster, protecting his meals and being forced into physical activity. His activity level increased significantly, to the point where he could even catch an entire rabbit on his own. Now, he's lived twice as long as snow leopards in the wild, defying all expectations and pushing the limits of his lifespan.
Like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.